Dear students, I am Ramesh, chemistry faculty from Vidya Mandir Metric Higher Secondary School, Uttangarai. Let us discuss medicinal chemistry. Now, what do you mean by drug? And drug is a French word. It is a French word, means herb. It is a dry herb. So, drug, which means dry herb. Drug is used to modify the physiological system or pathological state for the benefit of a recipient. So, drug which is used to modify the physiological system or the pathological state for the benefit of a recipient. And the drug will interact the biomolecules. They interact the biomolecule like lipids and enzymes. Lipids are proteins and enzymes, etc. So, they may interact with the biomolecule like that, the lipids and proteins and that enzymes. And the drug which cure the particular diseases, which is known as chemotherapy. So, what is that chemotherapy? Chemotherapy. The drug which is used to cure particular disease. So, the drug which is used to particular diseases, which is known as chemotherapy. And why we are using that? The drug uses, and we are using the drug for diagnosis. So, we are taking that diagnosis and prevention. It is prevention. And the third one, it is cure diseases. So, for that purpose, we are using the drugs. Now, ideal drug. So, the ideal drug is one which is the following characteristics. What are they? The first one, it is biocompatible. And it is biodegradable. And which is non-toxic. And which is no side effect. There is no side effect. So, these are the properties of an ideal drug. We are using most of the drug. We are taking most of the drug which has the property, which is biocompatible and biodegradable and non-toxic, and that there is no side effect, and it is only at low concentration. So only at a low concentration they are biocompatible and biodegradable and non-toxic, and there is no side effect. If it is a high concentration, then there will be a side effect. There is a side effect. Now the medicinal value of a drug. We are take medicinal value of a drug. So medicinal value of a drug. Medicinal value of a drug, which is known as, that is a therapeutic index. Therapeutic index. So by knowing that therapeutic index, we may identify the quality of a drug. And the therapeutic index is nothing but, it is the ratio of maximum tolerated dose of a drug. It is maximum tolerate dose of a drug by minimum curative dose of a drug. So it is minimum curative dose. Minimum curative dose. Then if maximum tolerative dose of a drug above which it has become toxic. Then if there is a minimum curative dose below which there is no effect on the biological system. So here, if the therapeutic index of a drug is maximum, then it is a ideal drug or the perfect drug. Now we are taking the classification of a drug. The drug is classified based on that properties. The first one, that is chemical structure. So we are take chemical structure. And the second one, that is a pharmacological effect. Pharmacological effect. Pharmacological effect. And the third one, that is action of drug. Action of drug. Then the fourth one, it is molecular target. Molecular target. So based on these, we are classifying the drugs into four types, that is chemical structure and pharmacological effect and action of drug and molecular target. So the first one we are take chemical structure. So we are taking the chemical structure. And in this classification, we are grouping the chemical structure in a single group. For example, we are taking penicillin. So here that is a penicillin. And the structure of penicillin is, we are taking here the penicillin structure, that is R, C, double bond O, N and H. And here there is double bond O and N, and we are taking here there is a hydrogen, and we are taking sulfur here, and we are take like this, and here it is CH3 group, and here it is a CH3 group, and here C double bond O and that OH group. Now here it is the general structure of penicillin. Now we are taking that here, depending upon the R group, there are varieties of penicillin. And if we are take the first one, here the R group may be, here it is C6H5, then it is CH2. R group may be, that is a benzyl group. Then if it is a benzyl group, then it is penicillin G. Penicillin G. So it is penicillin G. And here the R group may be, here that is C6H5OCH2. OCH2 group. Then it is penicillin V. Penicillin V. Then the third one, here we are taking that the R group, and the R group may be CHNH2. Then it is considered as, that is ampicillin. What is that? Ampicillin. So it is ampicillin. Then here the R group may be, here what is that? CH and NH2 group, and here there is a OH. Then it is known as amoxicillin. What is that? Amoxicillin. And the R group may be, we are take the R group, that is the fifth one. Here the R group may be, here the bond and CH3, and here there is a CH3, then it is considered as methicillin. So that it is methicillin. So they have similar structure and they are differing in that R group. So if they have the similar structure but they are different biological action. Likewise, we are taking the next structure, maybe opiates. What is that? Opiates. And also that is a catecholamines. 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 So depending on the chemical structure, we may classify the drug. Then we are taking that the second classification here, pharmacological effect. Now, depending upon this classification, the biological effect on the recipient. So, pharmacological effect is nothing but the biological effect on the recipient. Which is known as pharmacological effect. For example, we are taking that is antibiotics. Antibiotics. What do you mean by antibiotics? Which kill the bacteria or microorganism. So antibiotics are one which kill microorganism. Then they are known as antibiotics. And we are taking the next one, it is antipyretics. Antipyretics. Antipyretics are used which reduces fever. For example, we are using the paracetamol and Dolo 650, all are, it is antipyretics which reduces fever. So it action, what is the action? Which reduce fever. And we are taking the next one, anesthetic, which will produces loss of consciousness. So here it is anesthetics. Anesthetics are the drugs which is used to produce or induce loss of consciousness. Loss of consciousness. So, depending on that biological effect, we may classify like this. There are so many classifications. Then we are taking some example, that is antibiotics, antipyretics, anesthetics. Now, the third classification, it is a drug action. So, biological process we may classify, that is a drug action. And based on that biological process, we may classify the drugs into drug action. For example, we may take the streptomycin. Streptomycin. So, streptomycin, and also that is erythromycin. Erythromycin. And the streptomycin and erythromycin both are the drugs which is used to inhibit the protein synthesis on bacteria. So, both are used to inhibit protein synthesis in bacteria. But we are taking that the streptomycin. Here the mode of action on bacteria will be different. And the streptomycin which is inhibit the initiation of protein synthesis. So which inhibit or prevent the initiation of protein synthesis. But if we are taking that the erythromycin and it is the drug and which is inhibit or prevent incorporation of amino acid for protein synthesis. So which inhibit incorporation of amino acids for protein synthesis. So here generally both are the same drug which inhibit the protein on that bacteria, protein synthesis on that bacteria. But the mode of action is different. So streptomycin mode of action is inhibit the initiation of protein synthesis, but erythromycin which inhibit incorporation of amino acids on protein synthesis in bacteria. So different mode of action. So by that biological action on the drug we may classify drug action. And the fourth classification we are take that is molecular target. So we are take the fourth one, it is molecular target. And here we may take any drug which interact the biomolecule like lipids or proteins or enzymes. So we are taking that here the drug which interact the biomolecule like protein or lipids and enzymes. So based on this interaction we may classify the drug, and this is more specific. It is more specific. So here the molecular targets are more specific. So enzyme as a target. Suppose we are taking that the enzyme as a target. Enzyme as a target. And you know that enzymes are biocatalysts. So we are taking that enzymes are biocatalyst. And the enzyme action is more specific. So we are taking that the enzyme action is more specific. That means a particular chemical reaction, the particular enzyme is used. Now we are taking the enzyme DHPS. This is dihydroperoate synthase. Dihydroperoate synthase, which is known as DHPS. Then para amino benzoic acid. Para amino benzoic acid, which is considered as PABA. 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 Now, we are taking a drug that is sulfonylamide. We are taking the drug or medicine, it is sulfonylamide. We may take the mechanism of these enzyme and the PABA and the sulfonylamide. Now, we are taking the enzyme that is DHPS, that is dihydropyrovate synthase. And this DHPS as a active sites. We are known as the active sites. We are taking these two are active sites. Now, it has two active sites. Now, we may take the substrate, and the substrate we are taking that is a para-amino benzoic acid, PABA, which is known as para-amino benzoic acid. So, we are taking that the bacteria, and bacteria which converts PABA, that is para-amino benzoic acid, into folic acid. So, presence of folic acid, the bacterial growth is very high. So, the bacterial growth is very high only in presence of folic acid. So that the bacteria need a para-amino benzoic acid, that is PABA. So what will happen here? The PABA will attracted on that the enzyme, on that active sites. So it may be attracted on that active sites. It is nothing but the PABA. So here there is a PABA. So it is PABA, and here it is also PABA. Then what will happen? PABA, the bacteria which converts PABA into folic acid, so that the bacterial growth will be very high, and it will be affected the human body. Then what will happen if you are taking the sulfonylamide? You take sulfonylamide. So sulfonylamide is taken as a drug. Sulfonylamide is taken as a drug. Now we are taking the structure of sulfonylamide. Here C6H5, and here that is NH2, and S double bond O, and that NH2. And why we are taking the sulfonylamide? That means here para-amino benzoic acid, that is PABA, has a similar structure of sulfonylamide. So we are taking the PABA structure, para-amino benzoic acid. So we are taking that here the benzene, and here NH2. Both are similar structure, correct? Both are similar structure. Now para-amino benzoic acid, that is C double bond O, OH, and instead of C and here that is S and that NH2. So PABA and sulfonylamide has the similar structure. Now, if you are using the drug as a sulfonylamide, what will happen? Instead of PABA, sulfonylamide is attracted on that DHPS. The sulfonylamide attracted on that DHPS. So what will happen here? PABA is not attracted on that DHPS as a enzyme, so that sulfonylamide is attracted. So folic acid not produces. So here folic acid presence is reduces in presence of sulfonylamide. So here only PABA may be converted into folic acid. But what will happen? Instead of PABA, sulfanilamide may attracted or interacted with that DHPS, the enzyme. Then, so PABA is not converted into folic acid. So folic acid concentration is reduces in presence of sulfanilamide. So the folic acid is absent. So what will happen? The bacterial growth does not happen. So the bacterial may killed. So it is the mechanism of, that is enzyme as a target. Enzyme as a target. In some cases, the drug which attracted on the enzyme in a different site. So, drug may attached on enzyme with different site, not the active site. There may be a different site. Then what will happen? Here it will be attracted or binded with that different site. Then the active site structure is different. So the active site geometry is nothing but a shape. So active site geometry is different. This type of a drug, which is known as allosteric inhibitors. So allosteric inhibitors are one. That is, the drug may be bind with a different site on the enzyme, then it is known as allosteric site. So the geometry is different, so that the particular para-amino benzoic acid is not attached on that DHPS, so that we may get the allosteric inhibitors or allosteric drug. Now we are taking that the receptor as a target, and we are taking that the drug which bind that biomolecule, and these biomolecules are called as receptors. So receptors are the biomolecule. Now we may take that here the biomolecule or the receptor have that active site, and the active site is present on that outside of a cell membrane. So we are taking that the receptor. So here the receptor having that active site, and the active site is present in outside of the cell membrane. Now we may take that natural cell messenger is present, and this natural cell messenger is bind on active site of receptors, and it will be transfer the signal. So it will transfer the signal. Transfer the signals. So protein synthesis that will be done on that cell. Then what will happen here if you are taking a drug and which is called as antagonist. And antagonists are the drugs inside of the natural cell messenger. The active site is filled up by the drug that they are known as antagonist. So here instead of the natural cell messenger, the drug which bind on the active site, which bind on active site on the receptors, on the receptor, which is known as antagonist. Now we may take the another one type, that is agonist. Agonists are the mimic of natural messenger. So they are mimic of natural messenger. Natural messengers. So it will be give the different messages to the cell, so that these type of drugs, which is known as agonist. We are taking the example here. Adenosine is a receptor. We are taking the receptor is adenosine. So adenosine is a receptor. Now we are taking on the adenosine receptor here, on that adenosine receptor, adenosine is, we are taking this is adenosine. So adenosine is binded with that adenosine receptor. Then what will happen? It will produces or induces sleep. So when adenosine is present or bind on that adenosine receptor, will induce sleep. Now we are taking that the antagonist instead of adenosine. We are taking that instead of adenosine, we are taking the caffeine. We are taking caffeine. So caffeine is a drug which is used instead of adenosine. Then the caffeine will produces non-sleepiness. So here caffeine will produces non-sleepiness. So that the caffeine which is act as a antagonist. One, we are take morphine. Morphine is a drug which cause the neurotransmitter for the pain. So which stops neurotransmit for pain. So that here morphine may act as agonist. Morphine may act as agonist. So one is antagonist and that agonist. Agonists are mimic of a natural messenger, and antagonists are the one which block the natural messenger, then they will be bind on that receptors. Thank you.